Welcome to episode 19 of the Breaking Bio podcast. I'm your host, Stephen Hamlin from the University of New South Wales in Sydney, Australia. Today is February 21st, and we've got a full lineup today, so we're starting with Bug.
Hey, I'm Bug Girl.
From an undisclosed location.
Yes, my secret lair.
Hi, I'm Morgan Jackson. I'm a PhD student at the University of Guelph, Canada.
Hi, I'm Rafael Maya, PhD student at the University of Akron, Ohio.
Hi, I'm Tom Housley, a PhD student at the University of Stirling in Scotland.
And we're joined by a special guest today, David.
Hi, I'm David Winter, an evolutionary biologist without really having a job title at the moment at the University of Otago in New Zealand.
You may know him from Twitter as @theatavism, where you were one of the first people I actually talked to on Twitter, actually, when I first joined. You were the first person to actually reply to me. I was friendless and alone. But so moving on from my insecurities, we've got some breaking news to talk about this week, and that is the publication of the Sasquatch genome. And so we've brought David on in part because he said he thought about it for about 5 minutes. And well, it's actually almost a good business model. I mean, come up with something so ridiculous that people have to ridicule it, and then make them pay to ridicule it. You know, it's a good plan.
Wasn't there some question about the journal itself, that it had been recently purchased?
Yeah, so we should probably give a brief overview of actually what happened.
I was really hoping somebody was going to do that.
To get to where we're, to bring people up to speed. So actually there's this group of people in North America who are Sasquatch fans, I guess cryptozoologists, who have been going on for a couple of years about the fact that they've found genetic evidence that Bigfoot or Sasquatch is out there in North American forests running into out-of-focus photographs but never quite being captured or seen physically. And they've been going on and spruiking this paper, which is going to appear in Nature, is going to appear in Science, is going to appear somewhere, and it's never appeared. And then last week, it appeared in this journal called De Novo, which, or does someone else want to describe the web design of the De Novo journal?
I think Raphael had first-hand experience.
Yeah, it's a very flashy website with things that jump in your face and move around. And it's actually built using that Wix platform, you know, like the do-it-yourself in 15 minutes website thing.
Oh.
So it's GeoCities from the 1980s, is what you're saying?
Exactly, yeah. It's like the new GeoCities. It's really funny because like you can't really tell from the link, but when you click on the supplementary material, then it takes you to like wix.com/users. So it's like, oh my god, really?
Wow. So the point was that these guys, they gave up on getting it published in an existing peer-reviewed journal, and rather than doing what maybe if you really believe your answers and you can't get them through peer review, maybe one thing you would do is put them up on a preprint server and get people's feedback on them. Rather than doing that, they bought an existing journal, which I don't think anyone's been able to work out what the existing journal is, but it had sort of some sort of accreditation.
I did know that
one. They bought the rights to that existing journal, changed its name, built a pointy-clicky website in 15 minutes, and published their article, which is really something, and we'll talk about it in a second, I guess. And then put it behind a paywall for $30. So everyone that's read it, I guess, has paid $30 towards this research project, including Raphael.
Before we get into the actual article itself, I have to know is there a Craigslist for journals that you can go buy one? Like, what have I been missing all this time?
Yeah, exactly. I like how they took like the Bender way off, you know, like screw you guys, I'm gonna make my own theme park with blackjack and hookers.
With hookers and blackjack.
Yeah.
Well, that does sound like better peer review, but still. I mean, all right. So, for those of us who actually spent any part of our lives on this article, what thoughts do you have?
Well, I like the fact, I'm coming from a taxonomist point of view, and I do like the fact that they are trying to follow the zoological code for nomenclature, and they've actually tried to they have actually legitimately submitted the name, their subspecies name, Cognatus, to ZooBank, which is like the new repository for zoological names. So it has an LSID, so you can go through and you can find out what Homo sapiens cognatus is, and they've actually got a spelling mistake in their entry, so that's pretty good.
And I love it how they said that they couldn't deposit the sequences on GeneBank due to lack of taxon.
Yeah, so that's why they did the ZooBank thing. So now they can theoretically go and put them onto GenBank.
That's great. I mean,
I cannot wait for that.
Yeah, so you can find it on ZooBank, and yeah.
So that means though that name is actually now a name in good standing in taxonomy that exists, though, right? If they published it in a.
I would check the paper. Well, it's a subspecies, so it's different. But
Yeah.
I will double check that while we're talking. But my guess is that they may not have met all the requirements of the code, like a type and all that.
Oh, they probably don't have a type, would they?
Yeah.
Yeah.
If they had one, then this would be a really easy question to solve.
Exactly.
yeah.
You mean the blurry photo of the guy in the ape suit in the middle of the forest doesn't?
Yeah, they have like a video of a guy sleeping on a suit in the middle of the forest, and I'm like, you couldn't wait for him to wake up and film him afterwards.
I just love watching that and just imagining them going, "Oh my God, we should get some video of its face." No, it's sleeping.
I was going to say, some people who are listening to this may not know what a type specimen is.
Yeah.
Should we backtrack a little bit and explain the terminology?
Yeah, so a type specimen is like the flag holder for the species name, for not necessarily for the species, but for the actual name. So that's the specimen that name references. And so if you have any doubts about what that name might actually mean, you should be able to go back to the type specimen, the holotype, and compare anything else to it and find out whether it actually is Bigfoot or just a hairy pile of dirt.
The point of my bringing this up is that there is supposed to be an actual physical body somewhere that serves as, this is the type specimen.
Generally, yes, but there have been species described that don't have them. I'm pretty sure, actually, that human Homo sapiens doesn't have a type specimen.
I'm okay with that.
Yeah, and I mean, who wants to volunteer? I don't know, but
well, actually, sure, I'll do it. Sure.
There you go. But I think
it would be confusing people for centuries. It would be awesome.
Yeah, I think there was actually another recent thing where a monkey was described and there wasn't a type specimen put forward because it was like they only found like 4 of them in the wild, so they didn't want to kill one of them to make it a type. So as far as I know, those are the only ones that I'm aware of where there isn't an actual type specimen that's been designated.
Yeah, and I think in the bacteriological code, if you can't actually culture something, you don't have to have a type strain for it. So.
So they have a legitimate argument for saying, well, we don't have a type specimen because we can't find it in the woods.
Endangered. No, Sasquatch are endangered, man. We can't kill them.
Wow, that's a little hard to argue against, actually.
It's genius, really. The whole thing is genius. I mean, I'm just so mad I didn't think about putting stuff behind a paywall before this. I never think of these things until it's too late and somebody's already got them.
So I'm curious to what David thought about the actual genetics of the paper, because they do have long strings of letters.
So yeah, sorry. So actually, John Timmer, who is a scientific journalist at Ars Technica, and Carl Zimmer were trying to pull apart a little bit of what actually the genes that these guys report are. So in the paper, they distributed some genetic sequences, as Raphael says, long strings of A's and T's and C's and G's. And people that work in bioinformatics complain a lot about file formats and way that people distribute sequences, but I think in the entire history of bioinformatics, no one's ever distributed sequences as a PDF file before. But this is a giant 100-page long PDF of ATC, GC, GCT, GCGCA.
Wow.
So yeah. So I got my hands on these anyway. So I did have a bit of a play around to see what the sequences that they report were. So what they have is basically little piles of mud and hair, and in one case they have a downspout, like, piece of guttering, which is, they reckon, being chewed and has blood on it. And they reckon this is Sasquatch has come up to someone's house and eaten part of their guttering, and they swabbed some DNA off that as well. But anyway, they sequenced bajillions of these things, and they report whole genome sequences of 3 of them. So this is really quite expensive genetic work that I would wish I could do on my snails that I study. But these guys get to do it on hunks of hair and blood that they think come from a Sasquatch anyway. So they've got these huge sequences, and well, the first thing you would do if you're a geneticist is blast them. So there's this web tool, or you can actually run it on your computer locally as well, which just compares sequences to existing sequences. And when you publish in journals other than de novo any kind of genetic data, you have to put it into this big database called GenBank, which exists in the world and has information on all of the sequences that are published, and you can just basically compare it quickly and see what the sequences are. And when you do that, evidently, very quickly you realize that this is not a human, well, this is not a hominid sequence, right? Most of it, quite a lot of it, aligns to human chromosome 11. And why there are human chromosomes 11s walking around in the forest and not the rest of the human genome, I do not know. But anyway, it all aligns to human chromosome 11. And then, I don't know, 60% of it aligns to nothing on Earth. And the rest of it aligns to or matches all sorts of animals that you might expect to be in the forest. So you get lots of matches to dogs and cats, and actually one sequence, about 30% of it, the best matches to panda. Exactly. It's absolutely bizarre until you realize that panda is actually the only bear that's had its genome sequenced. So if you have large sections of the genome which won't be coming up in general studies that people do on bears, but will have been in the panda genome, well, that will be the best match. So presumably this is sort of bear DNA and all sorts of rubbish which has contaminated their sequences, and they just said, well, look, it's not human, so it must be, in their case what they say is it must be hybridization, where you must be looking at a hybrid hominid that moved into North America, I don't know, 20 million years ago when apes were sort of evolving.
I only scanned the paper, but that one line did stick out at me, this line they had about, they had 100% sequence homology with humans, but 0% with other apes, and Neanderthals and Denisovans, and
yeah. I mean, the whole thing is an advertisement for peer review and why peer review should stop stuff like this happening, basically, because these.
See, I want to believe that it's right? Like, I want to believe that there's this kind of panda bear composed mostly of dog and cat poop wandering around the forest. You know, I want to believe.
Oh, if only X-Files were still going.
Yeah, Mulder would be out there searching for this funky-ass panda bear.
You know, the whole thing just sounds like a furry gone horribly, horribly wrong.
Yes. It's like a fetish convention that just wandered into the forest and had a real problem.
Oh, dude, where's the con?
Yeah.
So to your best guess, you think they actually got a pile of fur and dirt and sequenced it, and not just went to GenBank and cut off sequences?
Yeah,
and put
so I
them
thought
together?
about that and wondered if it was. So we got two options at this point: either they're inept or they're fraudsters, right? And I think they're inept, basically, because, well, what I actually did is took, so lots of these things aligned, like I said, to human chromosome 11, and that's presumably because they saw lots of matches to chromosome 11 and then when they built their sequences. So these sequences are, like I said, they're from whole genome sequencing, and what you do when you use the modern whole genome sequencing methods, you sequence really, really, really small bits of DNA, about 300 base pairs or so, and then use computers to build them up into what the structure of it is, kind of like a jigsaw, I guess. And that's a much easier process if you have a real genome that you can build it against, rather than trying to build it up just by reference to all these little 300 base pair bits. So presumably they built their sequence by reference to chromosome 11, the human sequence, which you can go download and have a look at. So then I blasted their sequence against chromosome 11 in 50 base pair patches, basically. So took 50 base pairs of their sequence, compared it, took 50 base pairs of their sequence, the next one, and compared it. And there's like 300 base pair sections, or 50 to 300 base pair sections, which are perfect matches to human, and then it all just goes to crap. And that's the kind of pattern you'd expect to get if you had a big mixture of DNA. And some of it, you got the human sequence coming through, and some of it, they just mashed up with each other, all the different sequences that you've got, and the sequencer can't align it, and it kind of just makes a mess. And then some of it is the other sequences coming through is the biggest section. And those, they're just way too short to be the result of hybridization. So if you had hybridization, like they suggest was the reason for their the differences, you know, the sort of patchwork pattern that I'm talking about, you know, a section which is beautifully, perfectly aligned to human, then a section that's not. Well, you might expect that to happen by a hybridization, because you've got two different genomes, they come together, and then through recombination in every generation, chromosomes swap bits and pieces, and you get these hybrid chromosomes, right? But if you look at human genomes, if you look at human genomes outside of Africa, which is, we're all on this very, very white panel today, the result of this, well, we all have ancestors which left Africa about 200,000 years ago. But the pattern of leakage disequilibrium, this pattern of breaking up recombination, that results from that, because we have a smallish population size that left Africa, and then there's lots of us, and there's been lots of recombination. We still have patches of non-recombining sections which are about 20 or 30 kilobases long, so 20 or 30,000 bases long. And these patches in the Sasquatch genome are 50 base pairs long, so you know, it's just not plausible that this is the result of some ancient hybridization or something. This is messed up DNA, basically.
So they, in the paper, they go on and on and on about how they, you know, did this and that and the other thing to avoid contamination, and this isn't contamination, and we sent it to 7 different labs, and you know, they make a whole huge deal of it. So do you think they were just full of crap, or, I mean...
Well, I think what they should have done is looked at the ancient DNA people, right? So if a big part of your question is, could this be contamination, which is the obvious question that should bling in your head as soon as you see these sorts of patterns, well, then you should look to the field of science which is absolutely expert at dealing with contamination from small DNA samples, and that's ancient DNA. And ancient DNA can actually use these really high throughput next generation sequencing methods, because they've developed methods to deal with contamination, to measure contamination, to know when they have contamination and when they don't. And these guys just ignored all those, basically. So one of the points is that what they've tried to prove, another human species or a hybrid genome, those have both been done in recent years in real papers and real journals. It's not that it's impossible to do, it's just that they haven't done it, basically.
So all these researchers, if I'm not wrong, they're all forensic scientists, right?
Yeah, that's kind of terrifying.
Well, supposedly. Yeah, so that's my point, is if these are forensic scientists, have they actually, you know, convicted and people, or made people go innocent off of this science? Because I would bring pretty much any case that they have brought to court into question at this point.
Oh, yeah.
Yeah. Scary.
No, if I was a forensic geneticist, I would be doing my best to say that these people aren't part of my community, basically.
Yeah.
That is pretty horrifying, yeah.
Especially because they're from Texas, right? So I'm just going to bring it out that there's some really bad thing. We're making jokes, but this could actually have some pretty bad consequences.
Yeah. You know, I'm beginning to really doubt CSI right now.
This is what took you to doubt CSI?
It's a really good thing you're on a different continent, because otherwise I would totally smack you.
I mean, I thought it was a documentary, but maybe not.
Not so much.
The best explanation I can come up with is it's a viral marketing campaign for something. We're going to see a Sasquatch movie coming up soon or something like that. It's just it's insanely silly. When you see that video, you're like, you're not even trying hard.
It's going to be the worst Sasquatch movie when it turns out to be some kind of deformed panda thing in the forest.
It's actually just an hour and a half of the guy sleeping in the suit, right?
They didn't want to wake him.
Best movie ever.
One of the good things that happened from this, though, is that Joshua Drew, who goes by Labroides on Twitter, he started an absolutely awesome hashtag querying people for what issue 2 of De Novo Volume 1 was going to actually hold. And it spawned this huge meme for the evening until it got truncated by a meteor destroying Russia. But all sorts of great answers. And actually, Tommy Leung, who was on last week, just happened to come up with a quote that he just posted on our timeline. So here's his idea of what's going to be up next time. Human chromosome 11 as a selfish genetic element.
Wow.
But anyways, there's a huge...
Very good.
So anyways, there's a huge list of these awesome, equally ridiculous paper titles that are coming out soon in De Novo, I'm sure. And we'll put the link up on the blog, the Breaking Bio blog afterwards. But
I'm a little concerned that my chromosome 11 is walking around the forest without me.
and pooping everywhere.
Yeah. I mean, apparently it's acquired a body or something, eh?
It's 5 o'clock. Do you know where your chromosome 11 is?
Wow. Apparently it's all up in your sample, contaminating it. Well, we're actually coming up on half an hour here, and I felt kind of bad because we haven't talked much about what David does or anything. Well, so you're in between positions right now, right? So you just finished and you're looking for a postdoc or something?
Yeah, so a little bit of post-PhD, pre-postdoc limbo, I guess. So my partner is here finishing up her PhD in neuroscience. And while she's finishing up, I'm spending some of my time applying for postdocs and trying to convince people that they really need to have a postdoc they haven't advertised, but they really need me, which is... In Australia and New Zealand, this kind of stage is increasingly becoming a thing because we don't fund postdocs nearly to the extent that in the US and possibly in Europe systems do. So there's kind of almost... This last week there was a bit of a discussion on Twitter amongst scientists about the postdocopolis, which is the idea that in America it's relatively easy, not easy by any stretch of the imagination, but relatively easy to step from PhD to postdoc, but much, much harder to go postdoc to tenure track. And in New Zealand we kind of have that crunch down earlier because there's very little funding for postdocs, and the way the system works with overheads, they are charged a lot, so it's quite expensive for a PI to take one on.
Well, this is a really down note to end the podcast.
Yeah. Okay, so...
Can we go back to the cat and dog crap panda bear? Because that was a lot better than this.
In my internet trawling this afternoon, I stumbled across a website that was promoting new healthy treatments for dealing with stress and weight loss. The one that got my attention was the weight loss strategy of infecting yourself in the stomach region to get rid of fat with botflies. So I guess they're just proponents of you give yourself botflies and you lose your fat and it flies away with you, which is, I don't know, an absolutely horrible freaking idea that I can't imagine anybody taking serious, but I can almost see somebody trying it once.
So describe botflies for the crowd.
So botflies are these really cool, they are amazingly cool flies.
Cool.
Yeah, they're really diverse, and when they're larvae, they live inside mammals. So the human botfly actually lives in Central and South America, is where it's found, and it's got a really cool life history that I won't go into. But anyways, the end of the joke is that the larvae end up living inside your flesh, burrowing inside and sticking their ass out to breathe, and just eating you. And then once they're done, they'll pop out, pupate, and become an adult and fly off. But there's species that are in pretty much in all sorts of different mammals, from mice up to elephants. There's botflies for them. So essentially, what you're doing is you're getting your own little parasite friend that will wiggle around in your cutaneous, your skin and your subcutaneous fat, and eat you, and then fly away.
So the idea is that you'd have like a stomach full of fly larvae sticking out of your skin.
Yeah. When I say stomach,
that
I mean like
really
your midsection, because they're all on the outside. That's the thing.
Yeah.
They're all on the outside wiggling with their asses out, and it's really uncomfortable. I know people who have had them, and I'm hoping to get one myself one day, just as an experience.
Yeah.
But I don't want to intentionally inoculate myself with a belt of frickin' botflies around me.
See, I don't see this catching on, because that's really gonna fuck up my bikini photo shoot. Like,
oh yeah, totally.
That's a
Gary,
good point.
Totally.
But, if you do it low enough, it'll be below bikini line. So.
yeah.
I remember
What I'm
hearing
thinking. Go ahead.
Wasn't there like the tapeworm diet? Something
Yes.
like that? Like,
Hear about that every couple of years.
That's still around. It's still around.
Wow.
Should we give David the chance to pimp himself out for postdocs in case anyone with money is listening? They won't.
Yeah. You can record a little something, and we'll.
This is my elevator pitch, is it?
Oh yeah.
Yeah.
I don't know. Employ me. I'll. This is why I don't have a job.
Yeah, you might want to work on that elevator pitch, man.
I'll do
It's less of an
anything.
elevator, more of a free fall pitch.
Oh.
Well, what would your 30-second job application be, though?
My 30-second
Mine?
job application?
It's not an easy thing to put together.
That's true.
It sure isn't.
But no, seriously, man, I think you have great writing skills, and obviously I don't know that much about your molecular biology skills, but I'm sure they're mad molecular biology skills. But no, I mean, I think you can put together a great statement like that. So, rah, you can do it.
Cheers.
Well, there is truth to that. David, if you can't get employed, then the whole system
We're all
is just
doomed.
fucked. Like,
Yeah. Yeah,
yeah.
I agree.
We're just gonna all
He's a
pack up
very-
and go home.
He's a very talented guy. Someone should hire him. How's that?
Sounds pretty good.
All right. Well, then we'll put that as a tagline for this episode. Hire David. And
Sasquatch vigilante.
do you need a Scully to your Mulder? Here's your guy.
All right. Yes. There you go. That's your tagline.
Sadly, it's kind of true. The closest I am to famous in New Zealand, which is not very close at all, is for writing a blog post about this nutcase who thought he could predict the Christchurch earthquake, which we had a couple of years ago, which was very, very bad, by the moon, and that he was predicting another one was going to happen. So I wrote a blog post about this and ended up, because New Zealand is like this big, ended up on national TV talking about it. So that's as close as I am to famous in New Zealand, and every now and again I talk to someone who's like, you wrote that blog post, you're that guy. So yeah.
Kind of famous on the internet.
Very, very rarely I run into these people, but that is the closest I am to famous is for just pouring cold water on crazy people.
The way my sister explains it is, this is my sister. She's kind of a big deal on the internet. Which is, and then she has to add about pubic lice, and so that kind of spoils it. So I'd say be very happy your claim to fame is something legit.
All right, well, I got nothing to add to that. So thank you for joining us for episode 19 of the Breaking Bio podcast. And we'll see you all next time. Bye.